Good afternoon, everyone. I'm Kim Bischoff, the Executive Director of the Neurofibromatosis Network. Today, the NF Network is proud to present the third and final webinar in the 2025 research series. Stay in touch because we hope to bring you more of these cutting-edge webinars in 2026 as well. Today, we're going to hear from Dr. Angela Hirbe, who is an associate professor of medicine and pediatrics and director of the Adult Neurofibromatosis Clinical Program at Washington University School of Medicine. Her topic today is unraveling the impact of extra chromosomes, chromosome 8, on MPNST in NF1. Dr. Hirbe has received the second research grant that has been awarded from the NF Network Project DREAM program. This project is making great strides in funding innovative research through focus and high potential for the NF community. We will also be hearing from Dr. Moots. He's on the NF Tennessee board, also the National NF Network's board of directors. He's in neurology from Vanderbilt University, and the topic for his discussion today will be research trends in NF research and how you can influence them. And he's also the founder of this NF Network webinar research series. And then we'll finish our talk up today with Samantha Blaine. She's a patient education manager with Alexion Pharmaceuticals, and the topic of her talk is living with rare disease. And so I'd like to turn it over to you and introduce everybody to Dr. Angie Hirbe. Welcome.
Thank you. So these are my disclosures. So I'll try, I know that there are patients and patient advocates on this call as well as scientists. So I will try to explain things in a manner that I hope everybody can understand. We all know that NF1 is a complex neurogenetic disorder that really affects multiple organ systems. And, this slide just displays the diagnostic criteria that are used to give a diagnosis of NF1. And we know that NF1 can happen sporadically or can be inherited, through families. Just to show some pictures, right? So I think pictures are worth 1,000 words. So these are images of these things that are diagnostic criteria. So cafe au lait macules, neurofibromas, so the cutaneous neurofibromas or plexiform neurofibromas, the axillary or inguinal freckling that you can have, optic pathway gliomas, distinctive bone abnormalities, like this tibial dysplasia that you see here, and then the abnormalities in the eye, such as the Lisch nodules, these little benign nodules that you can see in the eye. I think it's also really important to keep in mind that different manifestations of NF1 happen at different time points throughout life. So things like the cafe au lait macules, the plexiform neurofibromas, the optic pathway tumors, those bone abnormalities, they happen early on. Speech and language delays and sort of motor and cognitive manifestations also happen early on. But things like cutaneous neurofibromas and MPNSTs, very scary sarcoma that patients with NF1 are at risk for, happen later. So really kind of late childhood, adolescence, and really kind of peaking that risk in adulthood. So just some basics, right? So DNA is the blueprint of life. It's the code that we all have. And this DNA gets wrapped up into chromosomes. And so we should have 2 copies of every chromosome in every cell in our body. And that DNA basically gets made into proteins. And proteins are like the doers, the worker bees in the cell. And this can get really complicated because these proteins interact with each other, tell the cell what to do through all of these very, very complicated interactions that I'm showing here. So NF1, is caused by a change or a mutation in the DNA. So this is a tumor predisposition syndrome where you have a mutation in the NF1 gene, which is located on chromosome 17. About 1 in 2,500 people worldwide are affected. As I said, involves numerous organ systems. So NF1, the gene, codes for that protein, the doer, neurofibromin. And neurofibromin is important because it's like a stop sign in our cells. It tells them to stop growing. So when you don't have this stop sign, this neurofibromin, then there are different pathways that are unregulated that are telling the cell to grow, such as the MEK pathway, AKT, and cyclic AMP. So we know that MPNSTs are the leading cause of cancer-related death in patients with NF1. And so this table lists a number of different tumors that patients with NF1 are at risk for, but the one that my group is really focused on studying are malignant peripheral nerve sheath tumors that happen in about 8 to 13% of patients with NF1. Unfortunately, most of the trials to date for targeted drugs for MPNST have been negative trials. So we haven't found any, really impactful, successful therapies. For these patients, most of them with disease that's already spread, that's meant that survival has been pretty poor. And if we look at something called progression-free survival, so how long does a patient stay on this therapy before their cancer starts to grow? In all of these trials, that average time is less than 2 months. So these therapies really haven't shown to be beneficial. What I think we've learned from the trials that we've done is we certainly have the ability to do MPNST only trials. But what I think is really important is that we start to incorporate biomarkers in our clinical trials. So we have some idea as to which patients are going to benefit from the therapies that we're trying. And so if we think about how an MPNST develops, a patient with NF1 in their Schwann cells, these are nerve cells, have one good copy of the NF1 gene and one bad copy. If they ended up getting another mutation, another mistake in the other copy of NF1, so you have no good copy of NF1, you end up with a plexiform neurofibroma. And then other genetic changes get you to MPNST. So losing CDKN2A gets you to this atypical state. And then other genes are lost that get you to MPNST. One of the other things that happens in MPNST is that we start seeing something called aneuploidy, where we get extra copies of chromosomes or we lose copies of chromosomes. And so my lab is really interested in using what we can understand from the genetic information from patient tumors and all of the mouse models that we've developed in the lab to understand what genetic events lead to MPNST, and then to use that information to try to develop better ways to diagnose MPNST early, and also to try to develop therapies based on stratifying patients into groups based on their genomics. And so one of the things that we've done in the lab is to develop what we call patient-derived xenografts. So these are basically when we take a tumor from a patient and we can grow it in a mouse, passage it in a mouse, and then test different therapies. And in doing this, we've looked at the genetics of the patient tumors and these patient-derived xenografts. And one thing that stood out to us was this identification that getting an extra copy of chromosome 8 seems to be a very common event. So if you look at this plot in panel C, this is basically a list of different patient tumors. And with each one, we have the actual patient tumor, and then below it, the patient-derived xenograft. And this plot is looking at every single chromosome that you have. And when you see blue, it means that the tumor lost the chromosome. And if you see red, it means it gained an extra copy of the chromosome. So what stood out to us was that chromosome 8 seems to happen pretty often. It's really the only recurrent copy number change that we see. So that got us thinking that this was very important for MPNST. And if we look at patients that have an extra copy of chromosome 8 versus patients that don't, that have the normal 2 copies, survival is worse for patients with that extra copy of chromosome 8, suggesting that it makes these tumors more aggressive. And so that was really the focus of the grant that we wrote through the NF network, was to really try to understand the mechanisms and therapeutic implications of getting an extra copy of chromosome 8 in MPNST. So we're looking at two things. One is trying to understand all of these pathways that are changing when you get an extra copy of chromosome 8. How does it affect how well the tumor cells divide and grow? How does it affect how well they survive? How does it affect the ability for them to make tumors? And then what are all those changes that happen in the proteins, right? The doers in the cell. And then can we learn more information to develop other therapies based on what we learn from this? And then also we wanted to start trying to look at different treatment responses based on whether or not a patient's tumor has chromosome 8. So this work has been done by some very talented postdoctoral fellows in the lab, Gorka Mastusam and Simgia Kar, who are both very interested still in NF1 research. So we found this mosaic population of human fibroblasts that we could purchase online. So meaning that these fibroblasts, which came from actual patients, some of them had the normal 2 copies of chromosome 8, but some of them had an extra copy of chromosome 8. Otherwise, the genetics of these cells are totally the same. So we basically sorted these 2 populations of cells and then worked with our stem cell core to reprogram them to be these pluripotent stem cells, meaning that they could become any type of cell that you want them to be. And we had these 2 groups, the normal 2 copies of chromosome 8 versus the group that has that extra copy of chromosome 8. And then we just proved that we had these 2 groups and they were what we think they are. This is something called a karyotype, where you look at what the chromosome content is in a cell. And like I said earlier, you should have 2 copies of every chromosome. So in that diploid group, that wild type group, we do have that. But in that trisomy or 3-copy group, we have that extra copy of chromosome 8. Then we use some other technology in the lab where we can edit in different mutations. And so we have our normal, our extra copy of chromosome 8. You can see 3 copies versus 2. And then in a subset of these cells, we then deleted NF1. So now we have extra copy of chromosome 8 plus you've lost NF1, or just the loss of NF1 group. And then we showed that we can actually differentiate these. So these stem cells can become any type of cell. We want to make them become Schwann cell precursors, because that's the type of cell that MPNSTs come from. And so this is what they look like growing as stem cells. And then in the middle, we can see what they look like in this middle kind of what they're called neurorosette stage. And then on the right, you can see what they look like when they're actually Schwann cell precursors. And so again, just a reminder, right, that different genetic changes we see in plexiform neurofibromas versus the atypical stage versus MPNST. And we're really trying to be able to model all of these stages and see what chromosome 8 does to the MPNSTs. So at this point, we have that, wild type, the normal 2 copies. We have the NF1 knocked out set, and that should make plexiform neurofibromas. We have that extra copy of chromosome 8 to see just what does chromosome 8 alone do to a Schwann cell precursor, and what does it do when you also have loss of NF1. But again, we need those other genetic changes. So we're working on making one where we have NF1 loss and CDKN2A/B loss, which that should make that atypical state. And then we think if you have no NF1, no CDKN2A or B, and chromosome 8, is that enough to get you an MPNST? That's one question we're trying to answer? Or do we need one of those other genetic changes as well? So right now we're working on making these other sets of cell lines. And then what we're going to do with all of them is look at how do the genes change? How does the protein change, those workers in the cell? How does it affect how the cells grow and survive? And how does it affect tumor formation? So Gorkham's actually made this, this cell line, which we think might make MPNSTs, and has shown that, yes, in the Schwann cells, or in the stem cells, they express high levels of stem cell markers. And when they're Schwann cells, they express high levels of the Schwann cell proteins and lower levels of the stem cell proteins. And so we have some preliminary data in looking at, how these different changes affect cell growth and survival. So what you can see here is that when you don't have NF1 or CDKN2A, and you have that extra copy of chromosome 8, the cells grow really well. So the higher the line goes up, the more the cells are growing. And then what's also interesting is that those wild type cells, now this is the opposite of what you see on the other side. Now we're measuring how much the cells die. So if they don't have chromosome 8 gain, they die much faster than if they do have chromosome 8 gain. Again, suggesting that it's important for these cells to grow and to survive. And again, I like pictures. So you can see here, we plated the same number of cells. So you can see just, very few cells in each of these different groups at the beginning of the experiment. But at the end of the experiment, you can see that the group that has NF1 lost and CDKN2A lost and that extra copy of chromosome 8 gain really grows the best. You have the most cells in this group compared to the others. And then in terms of survival now, when we see these bright green dots, it means that the cells are dying. And so in that wild type group that, has the normal number of chromosomes, those cells are dying faster than any of the other groups. And the group that's surviving the best here are the ones that have chromosome 8 gain. Again, suggesting that that extra copy of chromosome 8 helps these cells to survive better. Gorkham's also started to look preliminarily at how chromosome 8 gain the ability of chromosome 8 gain to allow the Schwann cell precursors to form tumors. And so he has taken those iPSCs and made them into the Schwann cell precursors and then injected them into the sciatic nerve of mice. And this is kind of where we are now. These experiments take a long time. So, he started this work 9-10 months ago. And we're still waiting to be able to look at the tumors under the microscope. But mice in these two groups have had to, we've had to put those mice to sleep because they developed tumors of a certain size that, we don't want the mice to be hurting. And so we're now in the process of analyzing those sciatic nerves, which look like they have tumors. And we think that these may be MPNST. And then this would be a model also for us to be able to test therapies. So we're next going to be, once we have all of these cell lines, our collaborators at PNNL, Sarah Goslin and her team, are going to help us look at these protein changes that are happening. So we hope to be able to do that in the second year of funding with this grant. But what we're ultimately interested in, right, are trying to understand how chromosome 8 is going to affect response to different treatments. And we know that the MEK pathway is important in MPNST because you don't have neurofibromin, and so this pathway is left unchecked. So we decided that we're going to use a MEK inhibitor as our backbone and then try to see what other drug should we combine with a MEK inhibitor. And we want to know, is there a drug that would work better in the cells that have chromosome 8 gain versus those that don't? And so We've done, you know, some fancy work with a lot of genomic data that exists to make these predictions. And what predictions came out is that we think that a drug class called an MDM2 inhibitor might work better in the cells that don't have an extra copy of chromosome 8. And a different drug class, something called a PLK inhibitor, that these would work better in the cells with chromosome 8 gain. And so again, Gorkum and Dana Borcherding, who's a senior scientist in the lab, have done some of this work. And so in terms of looking at whether or not the MDM2 inhibitor works better, these cells on this side have the normal number of chromosome 8, whereas these cells on this side have an extra copy of chromosome 8. And what you can see here is that this MDM2 inhibitor seems to be working better in the cells that have only 2 copies of chromosome 8 because they're not growing. They're staying relatively flat. Whereas over here, the cells are still growing despite being exposed to the drug. With the PLK inhibitor, it was actually predicted that cells would not just grow more slowly, but would actually die in response to having this drug. And that's what you can see here. So again, these are the cells that have chromosome 8 gain. So this experiment is kind of the opposite of the other one, right? So now we're looking at cell death. So if the curves are higher, it means the cells are dying more. And so we're seeing way more cell death in the cells that have chromosome 8 gain, where they're not dying in the cells that don't have an extra copy of chromosome 8. And so again, in the second year of our funding, we're really going to look at this and make sure it holds true inside an animal, so inside the mouse, and also combining with that MEK inhibitor, so the 2-drug therapy, not just the 1-drug therapy. At the same time, another scientist in the lab, Guangfeng, wanted to look at what are each of the individual genes on chromosome 8 doing and which ones are important. So she did this screen where she looked at all 652 genes and found that 58 of them seemed to be important for survival. And so she just looked at which one of these have drugs that might be targetable. And this protein PTK2, or this gene, sorry, the gene PTK2 encodes for the protein, the doer, called FAC. And we know that actually this pathway has been shown to potentially be important in MPNST before. And there's some interaction, again, with the MEK pathway. And so she did some very exciting work that showed when we give a FAC inhibitor along with a drug that hits MEK and RAF, in the cells, in the tumors with chromosome 8 gain, we actually see the tumors shrink away. So this is like something called a waterfall plot. So when the tumors grow, you see the lines going up, and when they shrink, you see them going down. And so this combination works much better in tumors that have chromosome 8 gain. So if we look at just the chromosome 8 normal MPNST, there's a little bit of benefit to the drug, but we don't make the tumor shrink away. And we have a grant from the Department of Defense to actually do the clinical trial, which was written by Jared Sears, who's a fellow in the lab. And this trial will open hopefully sometime in 2026 through the NF Clinical Trials Consortium, where we're going to look at this combination therapy in patients with MPNST. And then in the lab, we're going to be able to look and really try to prove that the group that has chromosome 8 gain does indeed respond better than the group that did not have chromosome 8 gain. Ultimately, what we want to be able to do is take the information from the patient tumors and in the lab figure out what treatments are going to work better for these different genomic groups of tumors so that we can really have personalized therapy. And my goal is to get us to the point where we have biomarker-driven clinical trials for MPNST. So I'll stop there and take any questions. But again, I want to thank all the patients and their families. I don't think there's a patient who's ever said no to contributing to the research that our lab does. Everybody in my lab who does the work and all of our collaborators, all of our funding sources. And I have to point out specifically Gorkum, who's done the lion's share of the work, who is amazing and is applying for pediatric residencies right now. And I hope he's going to stay in the NF field forever. And Dana, who's done a lot of the in vitro studies looking at cell growth in the Petri dishes, and then Guang Feng and Jared, who have been working on this FAC work and clinical trial. So I will stop and answer any questions that people have.
Well, thank you, Dr. Hirbe. It was a great presentation, and thanks. It's nice to see your team. You know, my daughter has NF, and I know many of the people that are on this call. It's really uplifting when we see, you know, a group of people like that all working on such an important issue. And there are a couple of questions in here that I will get to, but I do have one of my own. A mom that I'm very familiar with. She has a son that had an MPNST, but it was removed because it was caught very early, and he's doing well. So, do you know if there is, and this might not be in your wheelhouse, and I certainly understand that, but is he at a higher risk to developing other MPNSTs? Also, should he be checked to see if he has additional chromosome 8? Would that be something to help monitor where he is right now? Just would like to get your thoughts.
Yeah, of course. So I think we do think that if you've had one MPNST, there is a certainly probably an increased risk for developing another. I have a handful of patients that have had more than one MPNST, but I don't think we have a good sense of exactly what that risk is. In terms of how to measure chromosome 8 and should we be doing that? So I mean, right now, we don't have anything validated to say that it matters in patients. I hope that that's part of the data that's going to come out of the clinical trial that we're doing. The other thing is none of this work that I talk about today, but I have worked a lot with Taylor Sunby, Otto Chowdhury, and Jack Shern with CTF funded work to try to develop a blood test for early cancer detection. And that blood test is really based primarily on the fact that MPNSTs have that extra copy of chromosome 8 gain. And so that's actually something that we're working on validating right now through a test that could be run out of Mayo Clinic. And so I also hope that in 2026, we'll be able to open that test up to patients as an option to have done.
That would be great.
That would be free of charge through the grant for us to be able to get the data to ultimately get it approved by the FDA to then be something that insurance would cover. So again, I think our assay is pretty good, and we're working with Mayo to actually have the test in a clinical lab where we could do it for patients. So again, to be continued, but hopefully 2026 will be a good year for making progress.
Well, we'll be watching. Yeah, we'll be watching. So let me get to some of these questions. So I think I know the answer to them, and I'm going to let you do it. One of the person is asking, will this study be available in Toronto, Canada?
The
Yeah.
clinical trial for the FAC inhibitor and the MEK inhibitor will not be available in Canada. It's run through the NF Clinical Trials Consortium, so it would only be open at the sites through the consortium.
All right. And this one I think you addressed a little already. It says, how would we find out whether our child diagnosed with an MPNST has the extra chromosome 8? Is this readily available when working with our doctor team?
Not right now.
Yeah. Yeah. Okay. And then there was another question up here too that was talking about catatophin, wanting to know let me go back up here. What effect does catatophin have? I know that's not exactly what we were talking about yet, but that was Zachary York's question there. I don't know if you have the answer to that.
Where is the, oh,
It's in the chat.
yeah, I don't know.
Yeah.
I don't have a good answer for that.
Okay. Um, Well, and I think then somebody else is asking how they can volunteer. So I think we've got the questions here, Dr. Herbie. And if anybody else has questions and wants to put them in the chat, we'll collect them and send them your way if you don't mind.
Yes, that
Uh,
sounds great.
Great.
Perfect.
Well, thank you very much. It was a
Of
wonderful
course.
presentation. Best of luck to you and your team on this work.
Thank
We
you
hope
so
you have
much. We
great
appreciate
success.
your support.
Yeah, thank you. No, we'll switch over to Dr. Paul Moots, a dear friend of ours who has actually put together this webinar series. This was his idea to come up and do this year to talk about the different programs that have been funded through our Project Dream program. And he also is the president of the NF Tennessee group, one of the founders down there. They're running a wonderful um, advocacy program down there. They're member organizations of the NF Network. And Dr. Moose, you can go ahead and share your screen. You're muted right now, so if you're asking us a question, it's moving. While that's going on, it looks like we have a question here, Dr. Herbie, if you don't mind grabbing one more. Is there any thought or work through those therapies that can shrink peripheral nerve sheath tumors? Is there any carryover or possibility to use it and shrink inoperable brain or brainstem tumors or gliomas?
Yeah, that is an interesting question. I guess what I don't know is whether or not the drugs that we're using cross the blood-brain barrier. But certainly I would be open to, you know, this work being collaborated on. And so that's a question I'll reach out to the drug company to ask. Veristem Oncology is the one who makes these drugs. They're approved in ovarian cancer. They just got FDA approval last year for ovarian cancer. And they had reached out to us to talk about testing their drugs in our models. And it was sort of the same time that we realized that FAC was important. So it was sort of serendipity there that they reached out at the same time. But yeah, that's certainly something that I will look into, and I would defer to David Gutman on that one because I'm not the one who does the glioma research. But if they do cross the blood-brain barrier, then I will suggest that they perhaps look into that.
Yeah. Well, thank you. Thanks for the question, David.
Mhm.
All right. Well, we're going to go ahead and turn this over to you, Dr. Moots. Thank you very much for joining us today.
Well, thank you, Kim, for putting this all together, and thanks to everybody for coming to hear these discussions. And Dr. Irby, this is really wonderful to see and hear about, and oh, gee, to have people get a feel for what's happening in oncology and its relation to it. It was really great to hear, and appreciate you being involved with this. Uh, let's see. I put a little comment here to kind of a reminder to myself that these slides would be distributed. I think, in fact, they will be, this will likely end up as a webinar posted on the NF Network website. So maybe that's not quite accurate, but let's see now. Here we move ahead. Yeah, we've had now, this is the third of these webinars, as Kim mentioned, and there's been a little bit of some carryover from time to time in terms of discussions about clinical trials and participation and things like that. And these, I believe, are posted on the website. And if you want to go back and review them, there's some really wonderful discussions that are available, and a little bit of background to some of what I'll talk about today, too. So kind of as an overview, I'd like to kind of demonstrate what I think some of the trends have been in NF research, and then talk a little bit about patient quality of life and views, how those connect in with what's happening in the research realm, and how to try and contribute in a way to influence these things. So that's a little bit of getting started. We have found that in a survey we did with people in Tennessee, NF patients, that it was really important to them that there was NF research happening here in Tennessee, and there is, thank goodness. And I'm sure the people in Missouri are saying the same thing. But anyway, it's really one of the things that jumped out as very important for NF patients. And it's kind of, this is my way of thinking about it, but easy to intuitively understand that people look at this, they know these are illnesses or conditions with a lot of unknowns, and that these fields are moving forward, and to understand new developments and new trends is important. And to see new research provides a lot of hope that there'll be progress and a better life for people in the future because of all this work. So a lot of interest in this in the community, I think. One way to look at the trends in research is, who's publishing what and what are they publishing about? And this is a set of graphs or charts that come from the PubMed databases. It's a huge database of medical and health sciences related articles that is searchable, and it's available online. And I pulled out over a time course for NF1 and NF2 using the older terminology, and then for schwannomatosis at the bottom. And you can see the bars year by year. The scale is a bit different. It's a much larger scale for NF1 and less so for NF2 and even less for schwannomatosis, but all have this trend of increasing as years go by. And I want to point out around 1990, this is just after the NF1 gene was cloned, and quickly it was recognized that NF2 was a separate genetic disorder. And a few years later, the NF2 gene was then cloned and discovered. And this huge wave, here was the first wave, and coming right with it, this next additional upswing in research on these conditions. And I think of the trigger for that really being a lot of new information developing about the genetics and the spin-offs of those. The next time point I want to point out is around 2010 and soon after, when work on MEK inhibitors was evolving, and the trials for MEK inhibitors started to open 2014, 2000, soon in that time frame. And I think of this as the wave of oncology input into neurofibromatosis, you can see sort of at the same time the development of schwannomatosis information. And it was in this era that the separate genes that are now NF2 unrelated schwannomatosis were being reported on and discovered, and then a whole new trend or development there related to schwannomatosis. So a bit of background on that. And here I've kind of put out in numbers those same trends, a considerable increase in each of these areas of research. And to look at a broader view, if you look beyond oncology, you can see that by doing keyword searches, genetics, gene, imaging, QT, all of these areas have had a considerable increase in research interest and publications. And yet some of them, some learning, quality of life issues, pain, and these things have been considerably smaller in volume than the work on genetics and cancer. That's kind of been the first two waves of research as I see it, and these other things still are a little bit lagging behind. One other kind of background to this phenomenon, if you look at any field, and here I picked cancer and neurology and also anxiety and depression. Over the years, the expansion of research and publications has happened in all these fields, and there's more journals, there's more researchers, there's a lot of new information coming about, and so no matter what field you look at, there's going to be an increase across, pretty much any medical field in terms of publication. So the trends we see in NF aren't entirely related to internally what's happening in the field. To move on then, here's a look at clinical trials using the database clinicaltrials.gov, which I've talked about previously and is available. You can get online and look at this, and it's been modernized in a way that makes it more approachable for lay people to get information from, non-medical people. But anyway, this database has over 500,000 trials across all of medical fields, and really is a great source of information for finding trials. If you look, for example, for NF1 as a search, you find that there have been 253 trials registered in this national database. And currently, there are about 43 that are recruiting and some still under development. And you can do things, searches that, for example, look locally, like just at Tennessee, and see what trials are active in your area. And this is kind of useful, I think, for people locally to be able to go to. If you look at clinical trials, over segments starting in 5-year blocks, 2010 through 14, 15 through 19, 20 through 24, it gives you a little more of the trend of what's happening. And in this area, just after the MEK inhibitor trials are kind of concurrent with them developing, you see a really marked increase in trials for NF1. Some of the listings here include either NF2 patients for some reason, or were really mistakenly put on this NF1 category when they should have just been called NF2. So I've subtracted those out, but it's still the case that there's a considerable increase in these trials, and most are interventional or treatment trials. Some are observational, natural history studies and such, and there are a few registries. To look a little more deeply at that, in these same 5-year blocks, I've tried to map out, well, what are the trials aimed at treating? And again, you see oncology is a big part of this, and in this slide, that's kind of the focus of it. Malignant peripheral nerve sheath tumors and plexiform neurofibromas leads the list and is still increasing, and this wave continues, as you just heard. It had a big spin-off, I thought, in that looking at cutaneous neurofibromas, which had been kind of, everybody recognized as a hugely common issue, and yet without a lot of trials and work being done on it now in the last 10 and then 5 years, a considerable increase. And I think of that as a spillover or the effect of, success in treating plexiform neurofibromas, that now cutaneous neurofibromas, malignant peripheral nerve sheath tumors, and down the list, the common and important oncologic cancer complications of NF are getting more and more trials and more accomplished. So that's one of the big trends, I think. And yet, if you look at these same time frames, 5-year intervals for trials that are for NF1, but not specifically oncology related, what you see is something a little bit different, and I've here listed them: biomarkers, psychosocial, neurodevelopmental or developmental things, bones, scoliosis, registry, pain, etc. These trials have fallen off a little bit in terms of what's opening currently. Now, some of these are still underway, and there are some new ones coming up that are already in planning, but there's been this effect that some of the issues that are quality of life related haven't gotten the same groundswell or development. The oncology trials have really led the way by far. I think if you look at NF2 and schwannomatosis in clinicaltrials.gov, you find a similar kind of story in that, there's over many years now, 82 trials of about 14 that are actively recruiting, and one of which was available in Tennessee. When you look at the breakdown of studies, clinical trials for NF2 and schwannomatosis, the vast majority are oncology related, and those for psychosocial and other features considerably less common. Well, okay. So those are some of the trends that you can see in publications and in trials. And I wanted to also highlight what's in terms of funding, the trends that are going on. And this is from the CDMRP information that's online, and it shows that federal funding from the National Institutes of Health and CDMRP, the Department of Defense related funding for medical research, those two programs or funding sources have really been heavily the leaders in funding for NF research. And there are some other private foundation type fundings available. This got the most recent data that I saw, I was up to 2023, and of course in 24, 25, all of a sudden there's been a major shift in the availability of funds through CDMRP funding. And I'm not sure why they didn't update this further, although I would hope soon that for example, Project Dream starts to show up on this as that program grows. Here's another look at the federal funding from the NIH, which you can see is up to $40 million, really substantial, the amount that's been put into NF funding. And again, this is mirroring or reflecting, I think, a lot of what's happened in oncology and the great developments that's had in NF. This is sort of a similar diagram that's from the CDMRP, and it shows in the $15 to $20 million range, an incredible amount, and to lose or risk losing some of this funding would really be, and it has been a big setback for NF research. It shows here how some of this funding, or how this funding was divided up in terms of basic and clinical research. That's for the CDMRP awards. This also is for the CDMRP awards, and kind of shows that NF1, not surprisingly, takes the lion's share, or 2/3, I guess I should say, of the funding, and NF2 and combinations of that, and schwannomatosis for the rest. And in terms of the manifestations or problems that this funding is directed towards, again, oncology-related, tumor-related issues are 2/3 or thereabouts. Quality of life and cognitive issues, learning disabilities and things are considerably smaller, but an important part. So those are some of the trends that I think are worth looking back and seeing how they're developing. And I wanted to look then and kind of compare that to what people experience and in terms of their NF-related problems, how does that sync and match, or is there a mismatch? And one way to look at that is to look at guidelines for how to monitor patients. And there are guidelines that have been available for 30 years for children, for pediatrics, and there are now in recent years guidelines published for adults also. And I've kind of abstracted from those. But tumor and cancer-related problems are kind of the top of the list, or high on the list certainly, and neurological symptoms and complications. And then a series of other pain and depression and issues that are in the quality of life sector in a sense. And I must say, just as an aside, when physicians rate patient severity, and people have looked at this, and when patients rate themselves in terms of severity, like in quality of life scales, it's typical, it's been characteristic that the physicians see the patient's severity as greater than patients see it themselves. And so there isn't an exact match, but anyway, one way of looking at this is from the medical side. Tumor and cancer issues are some of the biggest problems, and it's certainly good that there's been so much focus and attention and development in the research on them. When you look at quality of life in NF1, there are some good scales that have been used. Dr. Ferner in England has been the champion of this for years, and she had patients do a 14 question scale rated between non and very severe symptoms on a 0 to 3 scale. Adding those points up produced this kind of graphic of about 50 patients, the majority of whom had kind of mild to moderate symptomatology, and not so many that were severe, really disabled by NF, but some. So to look at what patients thought were their biggest problems, what they saw as their outlook on life, struggles with depression and anxiety, pain and learning and those kinds of things. And it's a little bit the case that the current trends in research, those kinds of issues haven't exactly kept up with the oncology work. And yet it's also fair to say people who have certain medical problems, this kind of ranking and ordering may not register well. For example, if you had a really aggressive cancer, it certainly is going to have a bad impact on your quality of life. And yet you might not see it represented this way in this kind of assessment of quality of life. In NF2, there are similar scales of quality of life ratings and what problems impact quality of life. Again, Dr. Ferner had done this particular one, and it turns out for NF2, dizziness and hearing problems, which are directly the result of progressive tumor problems, rate the highest by far or considerably. It seems to me that it is in the NF2 where quality of life is more directly related to tumor impact, and in NF1 where there's this more diverse set of symptoms and set of problems, that direct connection isn't quite as set. So I've had these thoughts about these trends and how they impact on what patients experience, and it seems like to me there's the first wave was the genetics wave, and basic science issues and what was viewed as high medical severity issues kind of have led the way and have developed and been so successful that there's some spill-off and kind of thrust into other areas. And then oncology is kind of in that second wave, or in terms of new developments and new treatments, and also really very successful. And I have this sense, and I'm a neurologist, I should make that clear, that I think the next wave that gets at some of these other issues in terms of what you see listed as quality of life problems will come to better solutions, perhaps, or more knowledge and more understanding from the next, what I think of as the next big wave, which is going to be basic neuroscience research. I don't know, that's my speculation. And again, one side, last comment here is that with NF2, the oncology and the quality of life issues are more closely connected. And I think it makes a lot of sense that a lot of the research and clinical trials are directed in that way. So what can you do that has an impact as a patient or an advocate that has an impact on where NF research goes? And I think there are important ways, and participating and advocating and providing support for these activities in the long run will make a big difference. How eager are NF patients to involve themselves in research is a good question and has been addressed a little bit in some trials in psychosocial studies. Dr. Walters is the publisher of this one. But a lot of people who did this big survey said, almost 70%, that they would be interested in participating in a trial that addressed an important issue for them. And it didn't matter if it was randomized or a blinded study. Didn't matter if this was NF1 or schwannomatosis or male or female. So people said they would, and yet in that group, only about 6 or 7% did previously participate in a study. And there's a lot of good reasons. And in one of our prior talks, we talked about barriers to study involvement. Distance and cost and time. These are important factors for sure in what limit people participating. But what if you didn't have a problem with distance or cost or time away from home, and it only took 30 minutes a year? And that's kind of what the NF registry is. And it's still the case that only, I think, about 10%, certainly in Tennessee, it's about 10% of our individuals here, and I think if you look nationally, it's similar, participated in that. And it's a great opportunity to participate. So I wish we could boost up this 10% into the 60 or 70%. In terms of participating, it's good to know how to find trials, and a good way to start is asking your specialists, do they know about studies or registries or things like that? We talked in a previous webinar about searches, organized NF organization websites, clinicaltrials.gov and other databases. The Clinical Trials Consortium, which is a wonderful NF resource. It's the jewel of NF research, I think. And also looking at institutional trials, WashU, for example, Johns Hopkins, they have great websites that include their institutional trials that have been active. At NF Network, at the website, there's a page that's participate in research, and it includes trials that are looking to get patients. It includes a link to clinicaltrials.gov and to the NF Consortium, and also to Antidote, a database that will provide you with information about trials that are available for you. So that's a kind of a little bit more modern way of getting informed about what trials are active for your problems. Children's Tumor Foundation also has on their Life with NF page information about trial involvement, but they have particularly the NF registry, which is a great way to participate that takes very little really, and provides information about what kind of problems are important for NF patients, and people who are doing research or in pharmaceutical industry and other industries looking at that to see what do patients need and what do they want. And they also send you information about trials that are relevant for your problem. So that's good to know. I've mentioned earlier about clinicaltrials.gov, and for some people it's a little bit daunting because of the medical issue, discussions that they have, information they have. But they also have a lot of information about how to look and background information about the whole database. So it's good to look at, and it takes a few times to get used to it, but it's good information there. Two other databases that I think are very important and helpful, and one comes from Vanderbilt, remarkably, where they set up a database for clinical trials that patients could put in a little bit of information and get informed about trials for their medical problems, a way of connecting researchers with open trials. And there are a number of NF trials listed on there. These would also be on the clinicaltrials.gov website. Similarly, Antidote, which I mentioned before, on the NF Network website. Again, for me, I put in my age and location, and in terms of NF1 trials, looking for, they had 35 listed and 7 for NF. These also should be findable on clinicaltrials.gov, but here they will send you an update about trials that you might participate in. Another way to get more participation is to be able to do trials in a, decentralized is the term people like to use, a way where a trial, for example, at Mass General could be available across the country. And Dr. Vanessa Merker is one of the real champions of this, and making these trials available where people can involve themselves virtually, and really a big step forward for a community like ours where there are not a high number of trials, and people this way can participate without having to travel. Once more, to give a big kind of shout out appreciation for the Clinical Trials Consortium, which Dr. Hirbe had talked about. And this isn't quite the most up-to-date map of it, but I think there are now 24 sites, and heavily funded by CDMRP. They have completed, this was, I heard this fairly recently, I think 11 trials, and they have 4 or 5 that are supposed to open really by the end of this year. I'm not sure they're quite on track with that, but some really good, great trials. If you're able to participate in them, it's really super. Another way to participate is through the RAINS, Response Evaluation Neurofibromatosis and Schwannomatosis. This group of committees is geared towards putting together outcome measures for clinical trial design. What really would be a meaningful improvement in the status of my tumor, or in the status of my pain, or a whole host of things. And there are on every one of these committees patient representatives. And here too, Dr. Merker is a real leader in this, and so much so that she said, yeah, put my email address on there. If anybody wants to get involved, to let them contact me. So she's very active in that. So in terms of advocacy, the NF Network does a wonderful job in their advocacy program on Capitol Hill that runs yearly, and to support and encourage the legislature to fund through NIH and CDMRP NF research. And it's not the only way to advocate, but it's really a great one. And there are also important and valuable, research or legislation and advocacy issues that can be done locally at the state and local levels, but some of them have to do with insurance and coverage for research. They have to do with pharmaceutical companies having to develop trials for pediatric patients and things like that. So there's other things beyond just NF itself that has a big impact on our patients' ability to participate. And then at the local level, and I think people miss this opportunity sometimes, at Vanderbilt and most medical centers, there are patient advocates on their boards. And like our cancer center has a community board that includes patients. And to get interest in rare diseases and interest in navigators for NF or for young adults transitioning through from pediatric to adult care. These are places and ways that locally people can really raise the voice and raise the flag. So in terms of what you can do to provide support, I think there are a lot of ways to support people in the NF community, and everybody, I hope, finds their own way to do that. I would want to mention that in the United States, there's something about 120,000 individuals with NF, and if somehow every one of them gave $5, it would fund about 12 Project Dream Grant Awards. It's you might not think of it that way, but it's a community with a lot of power. And there's also within it the notion that if you get involved and participate in any of these ways, you get something back from it, maybe more than what you put in even. So anyway, just a few conclusions then. I think that what drives research, of course, at the fundamental level, it's patients and the problems that they face with NF. There are really a huge number of dedicated researchers. I mean, you listen to Dr. Hirbe and you hear the detail and the depth of thought that goes into some of these things. And clinicians and advocates really provide a lot to this community. Basic science has been, as I said, in genetics and oncology, really kind of forward thrusting, really important areas. And I think neuroscience is likely to become that also. Funding, of course, drives it, and research is a very expensive endeavor, and yet it's a very valuable one in its returns. And things that are successful, like the MEK inhibitors and like the oncology research, have really moved the field forward dramatically. So the success kind of creates kind of a snowball effect. How you can influence these things? The more you learn about research, the more likely you are to participate in it. And so sessions like this, learning is a good start, but there are simple things, registries and surveys and natural history studies, which are very valuable and easy to participate in. Clinical trials, the virtual ones are opening up more options. The trials that are clinical trials for treatment, they don't take hundreds of patients usually. It's 50 or, so it's not like everybody can be involved in those. But if you can, if that's what you need, it's really worthwhile, I think. Another thing we've tried to do, and I hope you all will do, is try and spread the word. If you know there's a trial open, tell the people you know in your own community who are friends with NF or colleagues or in your family. And then it's possible to get involved in trial design, another good way to participate. Advocacy is a real important thing, and find a way to contribute. And everybody, there's a possibility that they can find their own way, and I hope you do that. I'd like to thank Rachel Mindrup, who helped us in a little workshop create our own Find Your Own Journey painting. This was my effort at it. And but she's also graciously allowed me to use her portraits of many faces of NF in my talks, and I appreciate that. And so from our community, I hope some of you are out there listening. And for the wider community, thank you for listening in. And I hope these are some helpful ideas.
Well, thank you, Dr. Moose. That was a wonderful presentation. And I think Jason had asked the question a little bit earlier about how to find out about clinical trials, and I think you answered that very nicely for him. Thank you very much. Um, I truly appreciate you bringing these thoughts forward to everybody, about how they can participate because this community really does want to step out and help. I see it all the time. So thanks for reminding us and giving everybody the list of the different ways that people can participate and how effective that participation is. Let me see. I think there's one more question, Dr. Moots. Let's see. Oh, yeah, this one, I don't know that any of us can really answer this one. This one's from Scott. He said, one of the issues of participation is any compensation received is taxable and can negatively impact one's tax filing. Honestly, that's not the first time I've heard that question, but I don't know that we have the resources here, correct me if I'm wrong, to be able to answer that question for him.
Uh, not in any really knowledgeable way. It surprised me a little when I hear that, and yet I don't really know the details of why or what can be done about that. I wish there was legislation that made it possible to participate without there being any drawback.
Yep. Might be a good way for us to investigate that for next year. Scott, we'll keep that in mind as we're looking at the schedule for next year. Like I said, this has come up before. Um, all right. And then I think we're going to go ahead then. Thank you, Dr. Moots, and this will end the recording.